Hey guys, today we're going to talk about cellular respiration. So cellular respiration is starting with glucose and oxygen, and it's turning it into carbon dioxide and water. So it is the exact opposite of the photosynthesis reaction. So for photosynthesis, we are creating that glucose from the light energy from the sun. But in this case, we're not creating light energy, we're creating just energy in the form of ATP. Okay, so the summary photosynthesis equation was this, and we should know this from photosynthesis and think about, is this going to be endergonic or exergonic? So we should know it's endergonic because it's getting energy from the sun, so energy is going into the equation. And is it catabolic or anabolic? It's anabolic because it is creating something, it's building that glucose molecule. Okay, because remember, anabolic is where we're creating something larger. Cellular respiration is the exact opposite of our equation. So this time it is actually exergonic because it's creating energy. Energy is exiting the equation. And it's taking a large molecule and breaking it down into many carbon dioxide molecules. So it is catabolic. Okay, so what is the energy produced in cellular respiration used for? It is used to create ATP that can be used to fuel all sorts of things around the cell. And actually it makes like 36 ATP. And that ATP is used for all sorts of things, for creating different organelles, to just cellular functioning, to fueling active transport. All sorts of things require ATP. Okay, so this is showing our chloroplasts doing photosynthesis from our light. Okay, so what kind of cells even have chloroplasts? These are going to be like our plant cells or algae cells. What kind of cells have mitochondria? Plant cells and animal cells. Usually we think of animal cells having mitochondria, but plant cells also need to be able to take that glucose that was produced in photosynthesis and turn it into ATP. So all types of cells that are eukaryotic are going to have mitochondria. Okay, where do animals get their glucose from? So unlike plants where it makes their own glucose, animals get their glucose from eating food. So once we have that glucose, either for plants, it's already made in the cell. But for animals, how does that glucose get into the cells? Okay, so it's going to happen through transport, passive transport, specifically facilitated diffusion, because glucose is a large molecule. It needs some sort of transport protein to go through. Okay, so let's do a quick review of the structure of the mitochondria. We talked about this last unit, but it's got a double membrane. The first membrane is smooth and the second membrane is highly folded. These folds are called cristae. And that is where the electron transport chain occurs, which we'll talk about more later. Okay, and again, this is important because they're having a high surface area to volume, which means more electron transport chain, and then therefore more ATP can be produced. Okay, the space between the two membranes is called the intermembrane space. It's just a fluid-filled area. The matrix is the very inside part. Again, fluid-filled, and this is where we're going to have the Krebs cycle, also called the citric acid cycle, occur, as well as pyruvate oxidation. There's also, remember, DNA and ribosomes located inside the mitochondria, which comes out our idea of symbiosis, our endosymbiotic theory, where maybe these mitochondria came from prokaryotes. Okay, they also have their own enzymes that are all floating in the matrix, or some of them are bound to membranes. Okay, and different cells are going to have different functions, and some are going to have more mitochondria than others. So you can think about cells that have more mitochondria are going to have more energy needs, maybe like muscle cells or heart cells, things like that. It's a joke about the matrix. Okay. In our cellular respiration, we're going to have some electron carrier molecules. Okay. And you can see that we've already talked about some electron carriers in the past, but this ones are going to be NAD plus and NADH. And if you look, there's not a whole lot difference in their structure other than this hydrogen up here. We've added a hydrogen, but we've also added an electron because this is a positive and now it's neutral. Okay, we also have FAD and FADH2. Similar idea, it just actually is holding 2 electrons because it's got these hydrogens that are being added. Okay, this is a very, very similar role to NADPH in the photosynthesis reactions. Just remember that NADPH is used in photosynthesis. That P it doesn't stand for photosynthesis, but can help us remember that it's in photosynthesis. NADH and FADH2 are used in cellular respiration, but it's the same idea, is that they are carrying electrons. So they're just like little electron carriers. Okay, so our overview is that we are taking a glucose molecule, and over the course of many small reactions, we are breaking down that glucose. And those electrons from the breakdown of that glucose are shuttled by NADH down the electron transport chain. Okay, there are 4 stages: glycolysis, pyruvate oxidation, the Krebs cycle, also called the citric acid cycle, and then the electron transport chain. Okay, you can remember these by the acronym Good People Can Excel. Okay, sometimes stage 1 and stage 2 are combined because the pyruvate oxidation happens really quickly after glycolysis. So you can think of it as glycolysis and pyruvate oxidation, the Krebs cycle, step 2, and electron transport chain. I use these hand motions in regular biology to help us remember the steps too. So either one of these ways are ways that you can help remember the steps. Okay, step 1, glycolysis. This is actually occurring outside of the mitochondria. And this is important because remember, we also have prokaryotic cells that don't have mitochondria, but that means they can do this step. So glycolysis is going to occur outside of the mitochondria. Okay, and so what happens is we're just splitting glycolysis, lysing of the glucose. We're splitting the sugar, and it can occur with or without oxygen. And so it's believed to be pretty ancient, and that even early prokaryotes, because they don't need a mitochondria, were able to do this. And so it's going to take our glucose, which is a 6-carbon molecule, and turn it into 2 pyruvates, which are each 3-carbon molecules. Okay, when we do this, we're going to gain a little bit of ATP, and we're going to gain some NADH from those electrons that were lost when we split apart that glucose. Okay, this is actually a net gain because we actually have to use a little bit of ATP, and then we use a little, like we use 2 ATP, but we generate 4 ATP at the end. So it's a net gain of 2 ATP. Okay, so this is the idea. We're only making a small amount of ATP, so this is why those prokaryotes only use a small amount of energy, because all they can do is glycolysis. It also makes a little bit of water. Okay, that is our first step. Our second step is pyruvate oxidation. Those pyruvate molecules from glycolysis are going to go inside the mitochondria all the way into the mitochondrial matrix. Just remember that's in between, that's all the way past both of the membranes. Okay, and that occurs inside. Okay, so what's going to happen is those pyruvates are going to turn into a molecule called acetyl-CoA. Okay, so basically we're just attaching a molecule onto the end, and when that happens, we lose one of our carbons in the form of carbon dioxide. Okay, so it's a coenzyme, acetyl-CoA. I don't know that we really need to memorize that, but it we're losing 1 carbon in the process. And that happens to the other one as well. And remember, carbon dioxide is a small nonpolar molecule, so it can just go straight out through the mitochondria and exit the cell. Okay, so that's all that happens in pyruvate oxidation is the formation of acetyl-CoA, and we've lost now 2 carbon dioxides. The next part is the Krebs cycle, and it's going to occur also in the mitochondrial matrix. And we're going to take that acetyl-CoA, you can see it here in this first step. And we're not going to go through all of the intermediate steps, but the idea is that eventually it gets turned into a precursor called citrate, and we lose another carbon dioxide, so similar to what we just did before. and then we lose our other carbon dioxide. So all of our carbon is now being left as carbon dioxide. We've taken that glucose molecule and broken it off piece by piece. Okay, but when it does that, every time it's creating NADH and FADH2. Those electrons from that glucose is getting carried by NADH and FADH2. Okay, and that's the purpose of the citric acid cycle, is to crank out a bunch of NADH2 and NADH and FADH2. It produces a little bit of ATP, but we're going to get the majority of our ATP from our last step. Okay, so just as a review, we've made a little bit of ATP in glycolysis, and we've made a little bit of ATP now in the Krebs cycle or the citric acid cycle, but not a huge amount. The last step, electron transport chain, is where we're going to make the most of our ATP, and it's going to occur in the inner membrane of the mitochondria, and it's going to make around 32 ATP. As a review, where have we made FADH2 and NADH? We made NADH in glycolysis and in pyruvate oxidation. We made both in the Krebs cycle. Okay, so electron transport chain, all of this is now where we're using all those electron carriers that we produced in those first 3 steps. Okay, and this is also going to be facilitated similar to photosynthesis. We have hydrogens being pumped and creating a concentration gradient. So hydrogens are being pumped across the inner mitochondrial membrane to the intermembrane space. So we're pumping all of these hydrogens. So instead of being inside like the thylakoid, these are going in out of the matrix into the intermembrane space. This is the space between that smooth and folded membranes of the mitochondria. Okay, now that we've done that, we have a high acidic area in here, and these hydrogens don't like being here. They won't naturally diffuse. So they will diffuse through ATP synthase because of that concentration or proton gradient. And just like in photosynthesis, when they diffuse through ATP synthase, we are going to then convert ADP into ATP. Okay, and this is done by passing all these electrons down, similar to the photosystems in photosynthesis. Just these electrons are coming from NADH and FADH2. They go through the electron transport chain, and the final electron acceptor is oxygen. And so that's going to turn that oxygen into water. Okay, so just a little bit about electron carriers in the electron transport chain. So these are the electrons that were stripped away from the hydrogen of NADH and FADH2. They're passed to different proteins, and we don't need to know the names of these proteins at this point, but they're various electron carriers in that intermembrane, and each one is slightly more electronegative than the previous, meaning they're going to attract electrons even more and more to just carry it down this train until it gets to oxygen, which is the most electronegative. Okay, a little bit about ATP synthase. Remember, those hydrogens are going to go from high concentration to low concentration. That's in the form of diffusion. So it's a passive transport at this point. And when it does that, it's going to smash an ADP with a phosphate together to make ATP, and then we have ATP being produced. Yep. Another word for this is chemiosmosis. So it's the diffusion of ions across a membrane. So as those ions pass through the enzyme, part of the enzyme actually physically rotates, and that causes one part of the enzyme to shift into its active form so that ADP and phosphate can fit, and then they're joined together. So it's like they're physically being smashed together. So that is our last step, the electron transport chain. Again, some similarities between the electron transport chains in photosynthesis and cellular respiration is that we're using excited electrons to power the active transport of hydrogen into an area. In photosynthesis, that energy is coming from the sun and sunlight from those photosystems, but in cellular respiration, that energy is coming from those electrons from NADH and FADH2. We're also creating a proton gradient that is going to then be used to power ATP synthase. Differences, again, the source of those excited electrons. And in photosynthesis, we're producing ATP, and in cellular respiration, we are producing a little bit of ATP, but also we're using NADP plus or NADPH to produce NADP plus, and then in cellular respiration, we're using different electron acceptors. Okay, this is our review of cellular respiration. Bye, guys.